This will be a summary lesson on memory that assumes a general understanding of neuron structure and function, as well as episodic and semantic memories. We'll look at memory from three different perspectives: the system level, the allocation process, and plasticity between allocated neurons. At the system level, the idea is that a memory may be distributed across multiple brain regions. Within a brain region, we're going to have an allocation process. Only a subset of neurons will be responsible for storing the memory of a particular learning event. And at the level of individual neurons, again, an allocation process whereby only a subset of synapses on that particular neuron will be responsible for storing a memory. At the level of synapses, we'll see that the strengths of synaptic communication between allocated neurons can increase. We call this plasticity. So neurons that fire together will be wired together. And in order to make long-term changes to synaptic communication, the neuron will have to make proteins, and for that, then DNA will be accessed. DNA recipes to build proteins. The cell will make new proteins that will strengthen neural connections for long periods of time. Now, before we get to our story, well, let's lay down some basic vocabulary. Memory is the capacity of an organism to acquire, store, and recover information based on experience. And again, we'll make a distinction between two kinds of memory: episodic memories and semantic memories. Episodic memories include the circumstances of learning. There's a lot of context, a lot of detail. It's vivid. So the context includes you remember where you were when the event happened, when it happened, and lots of other details. We use phrases like "I remember" or "I can relive that event." So we can make a vivid reconstruction of some past episode. Perhaps you were in a classroom near Halloween, you were wearing your costume on the microscope, and you remember those details. Semantic memories are, they store just general information about the world, what it is that was learned on any particular episode of learning. We say things like, "I know about this or that." Perhaps on the microscope that day you learned about amoeba and how they eat paramecia. Now, oftentimes we have these episodic learning events, but we might forget the details. We might forget the context that was associated with that learning event, but we will retain what was learned. So this is an important relationship, that episodic memories provide the content for semantic memories. And oftentimes we do not remember the details or the context, the where and when we learn something, but we have that information stored. So that's our semantic memory. Now, the study of memory made great advances with patients like H.M. His name was Henry Mollison, and in the 50s he underwent an operation to treat his epilepsy. So as a young boy he suffered from severe epilepsy, and the surgeons at the time thought, well, perhaps they could just remove that bit of the brain that they thought was triggering the epileptic seizures. Unfortunately, the result of that surgery was that H.M. suffered profound amnesia. And so this gave scientists the opportunity to connect the removed brain tissue with memory function. Let's see what they removed from H.M.'s brain. Over here on the right we see the human brain. Up here we see the bottom of the human brain. So we've got the frontal lobes here and the temporal lobes here. So this would be the bottom part of the temporal cortex. And if we sort of cut top to bottom and look at a slice of the brain, here we see that section of the brain. And down here underneath the temporal lobe on both sides is a structure called the hippocampus. The surgery that H.M. underwent involved the removal of the bottom portion of the temporal cortex as well as the hippocampi on both sides. In this picture we see a whole brain slice. This is real brain tissue. There's the hippocampus. Notice we have the sort of gray areas. These would be where the neuron cell bodies are located, and then the white areas, these would be the axons. Over here we see the neurons have been stained with a purple dye, so we can see again the cell bodies in purple, and the white areas would be axons. We'll use this diagram to describe H.M.'s amnesia. The arrow would indicate the time at which he had the surgery. After the surgery, H.M. could not make new episodic memories. And as a consequence of that, neither was he able to build up new information about the world. He couldn't make new semantic memories. That's a profound memory disturbance. Now, other tests revealed that HM could learn new motor skills, and that showed that memory is not a unitary phenomenon. There are multiple memory systems in the brain. HM did have good recall for semantic memory of the remote kind. So he could describe his childhood, and he had basic facts about the world. And of course, the fact that he could carry on a conversation means that he had stored meanings of words in the English language. So he had good recall for remote semantic memories. Well, this suggested then that the hippocampus is necessary for making new memories and somehow transforming them into a long-term system that does not require the hippocampus anymore for retrieval. In fact, HM showed a gradient of memory recall. Events just before the surgery, he had poor recall, but as scientists asked him about events more remote in time, his retrieval got better and better. And so it was as if it takes time for the brain to transform a recent memory into a remote memory that can be retrieved in a different way without the hippocampus. In the next part, then, we'll try to get to the bottom of why is the hippocampus so important for memory processing.